Well, good afternoon, folks. Good afternoon. Welcome to the Jones Seminar. Today it's a pleasure to have Jessica Lasky-Su give us a talk. She is an assistant professor of medicine in statistics at the Harvard Med School, having done her bachelor's, she was telling me, at Cornell, and PhD at the Harvard School of Public Health. It sounds like stayed there ever since, which seems like a good place to be. She's associate statistician in the Channing Division of Network Medicine at Brigham Women's Hospital. For the past 15 years, she has experience in the analysis of complex data sets, particularly genetic, genomic, and metabolomic data. She has over 100 research articles within many areas of genomic and vitamin D publications. She's now working on R1-funded research around metabolomic data to identify the metabolic profile that predicts asthma exacerbations using two asthmatic cohorts. She works with collaborators Weiss and Letonia, the principal investigators on a large national vitamin D antenatal asthma reduction trial called VDART. VDART is a randomized, double-blind, placebo-controlled trial of vitamin D supplements in pregnant women to determine whether prenatal supplementation can prevent the development of asthma and allergies. So she'll be talking about some of her work in statistics and the context of that. I think probably most notably you might recognize the last name, and I have to recognize that she's also the daughter of Professor Ron Lasky here, who probably half of this room has taken statistics with, so statistics seems to be in the blood. So please welcome Jessica today.
Thank you so much for having me. I want to preface this talk by saying I'm really excited about this talk because it's such a contrast to what I really, to a lot of the talks that I do that are very honed in on my specific research. But this talk is designed to take a step back and look at really how statistics plays out in medicine and how us as a public look at those data and look at those outcomes and determine what the data are telling us on a sort of mass level. So with that, the title of my talk is Statistics in Medicine, Why the Data Tell Us One Thing One Day and Another Thing the Next. I want to talk this, just go over what I'm going to say here. I'm first going to just talk about what my job is as sort of a medical research professor and some of the really important aspects of being successful in that as a position. I'm going to go over really what the requirements are, I think, and what some of the challenges we have as health researchers, and I'm going to summarize some aspects of my research. I'm then going to totally switch gears and go into what this talk is about, which is really statistics in the news and how we interpret that. And I'm going to give 4 different examples that I thought were relevant from various degrees, including an example on autism, vitamin D, hormone replacement therapy, and running. And then I'm going to have some conclusions. So with that, let's just talk about what it's like to be an applied statistician, principal investigator at a medical school. Unlike here at Dartmouth, we are not funded on hard money, which means 100% of my salary is covered by writing grants and doing research. So people who we love and are near and dear to our heart include the NIH, who has $31 billion to help fund us with our research, as well as private research centers. The focus of a lot of what we do is doing research and writing manuscripts. I do a lot of attending meetings and leading meetings where a lot of critical thinking and discussion happens about different analytical approaches we're going to take to our research. These different meetings are always multidisciplinary in nature. In contrast to a setting that is a university, instead of having a lot of doctoral students, we have a lot of postdoctoral students. So if any of you out there are interested in looking for a great place to do a postdoc, come see me after. I also give a lot of talks. I was just at the NIH yesterday giving a talk to the leaders in the NIH. Of course, we do data analysis and also some level of teaching. So let's just dive into that a little bit further. Here is an example of what I mean by interdisciplinary collaborations. These are all some of my close colleagues. Here is Dr. Weiss, who is really my main mentor for the last 10 years. He's an asthma MD doctor with over 800 publications, 69 and still going strong in research. Here's another one of my long-term collaborators, a biostatistician who's really focused his whole career on looking at the genetics of complex diseases and developing statistical methods of that. Here is someone who is near and dear to my heart. This is my postdoc, Rachel, one of my postdocs right now, who is this cute little English girl who also does a ton of work in the nitty-gritty with programming. She's an epidemiologist. Here is one of the research teams I lead, where we have bioinformaticians, MDs in asthma, MDs in allergy, other epidemiologists, some focused on interesting things like the microbiome. Here we have someone who is more of a pathologist, a chemist, another general MD, but also who focuses on metabolomics. And here is one of our pharmacology colleagues, and here I am leading this group. So interdisciplinary collaboration, not only at the level of diversity of background, but of diversity of where we're located. I have all these great collaborators around the world, most notably Europe, who are really fundamental to my research success. Another aspect I love to point out about research, and I think this is something especially doctoral students need to recognize, that I was sitting down one day with my boss, and he looks at me and he says to me, Jessica, you know, when I look at the professors at Harvard, they're all smart. They all have IQs of about 120 or up. But when you look at that group, it's not always the smartest people who are the most successful. Then he says, I'm not that smart. I have maybe an average IQ for Harvard professors, but I've been incredibly successful, and one of the primary reasons is because I have good ideas and I can think out of the box. And I've really found this to be true. It's actually striking that some of the smartest people have difficulty succeeding because they don't have these other important attributes of working hard, having good idea, persistence in getting funding when people tell you, no, your ideas are not good, and also social intelligence, being able to interact on multiple levels with an array of people. Now, I found one thing very interesting is that Malcolm Gladwell also echoed the same comment in his book Outliers when he commented that the relationship between success and IQ works only up to a point. When someone reaches an IQ of somewhere around 120, having additional IQ points doesn't really seem to translate into any measurable real-world advantage. And I've really found that to be true in my 15 years of research. Now, other issues I face in my research, one is something that the genetics and genomics community has been facing in the last 10 years, and that is the overwhelming amount of data that we now get. So I was just at a meeting yesterday that was at the National Heart and Lung Blood part of the NIH, where they talked about this group of researchers, and we have now gotten the sequence data on all of these individuals. And one of the people pointed out that the data that we received was on the order of 2 times 10 to the 15th measurements of genetic data points. And then they said, to put that in perspective, that is almost double the corn production that the U.S. has in 1 year. So when it comes to challenges on a statistical level and an analytic level, this is one of the first things we face is the overwhelming amount of data. And in fact, over the last 10 years, this number has gone up exponentially. So now going on to a little bit of what I do. I primarily focus on a disease called asthma, which most of you know because it has a very high public health impact and prevalence. I assume that there's a good number of asthmatics in this room. Some of the primary characteristics of this disease is inflammation of the airways that results in difficulty breathing. Looking at this on an overview level, one of the things I focus on my research is what I call the Ohmic Hierarchy. So for those of you who've taken some biology, one of the things that we know is this relationship between genetics go to DNA, goes to RNA, which goes to protein, which goes into different metabolites in our body. So what we tend to do is we get information on all of these different levels, in addition to all the different environmental exposures someone may have. So let's take a person, we get measurements of their DNA, their RNA, their metabolites, and environmental exposures. So for asthma, important things like maybe dust around the house, exposure to dogs, exposure to foods that you may be allergic to. And we look and try to see how all of those interact to develop a disease. Now, I primarily focus on two different things right now, which is genetics and metabolomics. So when you look at this slide, really two levels of this and this. Primarily because when we want to look and connect genetic components to a disease, the metabolites really give us this great measurement of the disease pathogenesis. And what do I mean when I say metabolites? So metabolites are measures in your blood or measures in a tissue. So let me give you some examples. Glucose level in your blood. This is a level that actually is part of the definition of diabetes, but it is also a metabolite, and it can be measured in the blood. Triglycerides, so when you get measurements of cholesterol and you have HDL and LDL cholesterol, those are both different metabolites. Now when I give these examples, you can see how these metabolites are inherently related to disease. So if we can see how those metabolites are being formed through environmental influences and underlying genetics, we can really get an idea of disease pathogenesis. So just thinking of this on a global level too, one of the primary analytic tools that we use to do this is something called networks. Now when you think of networks, you can think of networks in a variety of concepts. One of the greatest networks I think of is the World Wide Web. When you think of the World Wide Web, you can think of every time you click on a link, there's connections to other websites. Now there's some websites that are highly, highly connected, whereas other websites aren't highly connected. So, for example, Google has a few more connections than maybe my personal website. So let's say you took the internet and you said, I want to get rid of this internet. It's a bad thing. What would you do? Where would you go to attack? Where do you think you'd go?
The largest nodes or shut down all the servers and wires.
Absolutely. So you're going to attack Google, you're going to attack Amazon, you're going to attack those eBay, you're going to attack the major hubs within the network. So translating this easily to, if you think of plane courses, you may think of things like Chicago and New York and LA. But there are also biological networks, and here's an example of a biological network where you have these nodes that are highly connected, also called hubs. The key thing when we think of disease pathogenesis is if you destroy those hubs, you destroy the network, and thereby possibly destroying the disease. And so that is one of the goals of what we do. And we can generate networks on multiple levels. We can generate genetic networks, we can generate metabolomic networks. We can also see how these networks interact. And so I spent a lot of time working actually with theoretical physicists who are really experts in network theory to help do this. And here's an example of a network where we have genetic data and this metabolomic data, and we've developed a network for asthma. Now, just to give you some idea of the types of things we can extract from this network, one of the metabolites that we identified in this network is this metabolite called sphingosphene-1-phosphate. And it's the crucial metabolite in this network for asthma. Now, connecting that to genetics, I'm now going to show you some maps of some different genetic regions. This region is with some of my own data, but it highlights this particular part of chromosome 17 that has 3 genes in it. One of them being this gene right here, ORMDL, sphingolipid biosynthesis regulator 3. Wait a second. What did I say here? Sphingosphene-1-phosphate. Sphingolipid biosynthesis regulator 3. And yes, those two are related. Now, more so in my field, this actually turns out to be the #1 replicated region associated with asthma that has been identified to date. And this was one of the large-scale studies in the New England Journal of Medicine, one of the most prominent medical journals, identifying the same region. Now, our group has gone on to look at this region even more, and we've actually developed a knockout mouse where this gene is actually knocked out in the mouse. And strikingly, you know what the most difficult thing of doing this research with this knockout mice is? They have trouble breathing. So this is a good thing for us because it really shows that ORMDL3, which before all of these genetic studies were done was not even thought of to be related to asthma, really showing us that there's something here. And really bringing this one step further to the clinic, one thing we found is that this sphingosine 1-phosphate is formed by sphingosine kinases that have identified possible drug targets for the treatment of asthma. So this is actually suggesting that they're actually therapeutic targets that we can now target based on this genetic and metabolomic research that used networks and different genetic and metabolomic analyses. So this is a summary of the type of research I do, and this is where I'm going to stop that part of the lecture. Other than one last thing, and I think this is a crucial thing when you're doing research and talking about motivation, and that is this picture here. So this little guy is one of my 4 children. Gulp, yes, I have 4 children. And this is him in November 2014, and this is him in November 2015. He has one of the highest measures of IgE, which is a measure of allergic reactivity I have ever seen in my life. And bringing him to Children's Hospital, where we've talked with allergists, and knowing I work with allergists from Children's Hospital in Boston, they've all said the same thing. So when I think about my research and when you think about your research, I just encourage you to have something that motivates you to do your research. And I always think back to him as one of my primary motivators, and I'm happy to say for the last year, this is how he's looked instead of this. So moving on to my main talk today and looking at statistics broadly in the news and health research. One of the things that always has boggled me is when, before I had children, I could actually listen to the news. I would sit down and I would be listening to one of these people, whether it's Brian Williams, Katie Couric, or someone of the sort. And they always have these things. A new study has found that. A new study has found that drinking a glass of wine a day is good for you. You know, bacon is actually good for you. Butter may not be bad after all. Statins may not be good for you. All these things that sort of contradict what we may be thinking, and which I sort of think back and I'm like, wait, 2 years ago you said the opposite. What's happening? And it really begs this question. Why do findings in health research change? Now I'm going to talk about this using 4 different examples that I think all illustrate different points. Autism, vitamin D and asthma, hormone replacement therapy, and running. And in each one of these examples, I'm going to show it from a little bit of a different angle. Now, the biggest issue with this has absolutely been the studies of autism and linking that to vaccines. So the year is 1998, and we turn on the news, and we all of a sudden see the news forecasters saying, vaccines cause autism. And it all stemmed down to this paper that was in the Lancet, a very high-profile British journal, and this research by a fellow named Dr. Andrew Wakefield. Now, the title of this talk is actually doesn't really make sense for people sitting in this audience probably, and doesn't actually really make sense for me until I looked at it further. But basically what he's doing is he's saying that a common vaccination called MMR, measles, mumps, and rubella, when given to children, caused this particular type of autism that also had colitis, which is an inflammation of the colon. And this paper then all of a sudden became, people just went crazy with it, and all of a sudden we see these things. You know, mercury poisoning and autism, these dramatic pictures of vaccines being shoved in kids, and if you just give us a safe vaccine, we'll use it. You know, we shouldn't have to decide between polio and autism, and so on and so forth. And this just created this reaction that ended up causing all of these myths. Vaccines are ridden with toxic chemicals. Well, vaccines, if I don't vaccinate my child, it's only going to affect my child. I should have, the say in this. When you give all these vaccines, it's going to overwhelm my child's immune system. And it's just the drug companies. They just want more money. So this is a type of reaction we got, and it's been a very, very passionate reaction by the public. But I think it's important to look back a little bit more critically at this initial study. And when you do, you might be incredibly surprised. So you might be surprised to realize this initial study was based on a whopping 12 people. Okay? This was not based on any statistics. There was no control group. And basically, these people had a retrospective recollection of what happened. And they were just called on the phone, and it was like the mom saying, yeah, when Johnny got that vaccine, he started acting a little weird. And I think he has autism. So it wasn't even based on a medical diagnosis. So this, to me, is pretty much almost criminal when it comes to peer review. Because peer review of scientific research is really necessary for the public to get the right view. Okay, so that study was pretty bad, but at least the scientific reaction to this was very good. So this study was followed up in 1999 by a study of 500 children, in 2001 by a study of 10,000, 537,000, 535,000, 10 million, 14 million individuals, and all of these studies were well designed with control groups and statistics, and they all said one thing. Conclusively, there is no link between autism and vaccination. As a result, The Lancet retracted this article. And ever since, we have been trying to get out one message, and that is, how do we know vaccines do not cause autism? The answer is science. Science suggests that it doesn't. However, still, we have these thoughts that have infiltrated our thinking, where 1 in 4 people think that autism is caused by vaccination, and a lot of people choose to not vaccinate their children. Now, I'm going to get onto this a little bit more, but I think one of the key things we have to realize is when we decide not to vaccinate, it doesn't just affect us. And I'm going to hold on to that thought, where I just basically want to say that vaccines do work, and they've had tremendous public health impact. They've eradicated diseases like smallpox, they've saved lives, and they significantly have reduced the disease around the world. So some of our conclusions here are that the initial science was not valid, and therefore the conclusions were not correct. But they have had this strong public health impact and have had bad ramifications. And the thing that I think threatens us this most is that one concept that is called herd immunity. And that is illustrated here, where let's say you have a few people in red who have a disease, but if the population as a whole is not vaccinated, then the entire population ends up having the disease. There's a critical threshold whereby if enough people are vaccinated, you will have herd immunity, and that means the disease will not spread in the population. So we really need to think about the fact that most of us need to be vaccinated in order to prevent these huge outbreaks of disease. So in summary of this first scientific issue was autism and vaccine. Why do scientific findings change? Answer number 1, bad science. Going on to the second topic, and this is one that I'm going to show you, and this is actually some research that's done in our group is going to be presented here, and actually this research that I'm presenting is hot off the press and actually going to be published next week in the Journal of American Medical Association. So please don't, I'm presenting it a couple days early, but I think I'm okay here. And this question is, vitamin D, A lot of people have a lot of thoughts about vitamins, but does vitamin D supplementation affect asthma? And this has become a really popular topic over the last few years, and here are some papers. Vitamin D supplements do not improve symptoms or treatment. Low vitamin D tied to asthma attacks. So right off the bat, we're asking this question, what happens? Does vitamin D have an effect? And why even would it? What do we even know about vitamin D? Well, vitamin D is something that's found only a small amount in foods, although now we are fortifying our food, so we are getting more vitamin D from food. But the key way we get it is through sun exposure and taking vitamin D supplements. But one of the critical things here is in the last 50 years, people in especially first world countries have progressively become deficient in vitamin D. I, however, am the poster child of someone who is not vitamin D deficient because I am small and I have very pale skin. And I run a lot, so I'm out in the sun a lot. So I actually have a great vitamin D level. But a lot of people don't fit that description. So here are some shocking numbers on the prevalence of vitamin D deficiency, and I will say I actually think this is an underestimate of the numbers. And when you see here, they're roughly 20 to 30%, and I would actually say it's over 30% of our population is vitamin D deficient. Okay, question. This has happened over the last 50 years. Can one person give me a reason why they think this has happened in the last 50 years? Sunscreen, on the nose. Sunscreen, we've become much more careful about putting on sunscreen to prevent diseases like cancer and skin cancer, and that has also helped us absorb, with absorbing vitamin D because we're protecting our skin against it. Other reasons, we're inside a heck of a lot more now than when we used to farm outside, and our lives are more sedentary and indoor. You know, my kids, kids playing video games rather than running around, et cetera, et cetera. Now, why would this affect asthma? Well, one of the key ways that it's known to affect asthma is because vitamin D plays this critical role in immune response that prevents inflammation. And as I said earlier, asthma is an inflammatory disease. And here are a bunch of my friends. They aren't actually my friends. These are all ways that inflammation happens in asthma. And so the biological link is very plausible for vitamin D impacting asthma. So I'm now going to talk to you about two randomized clinical trials that have just been performed in vitamin D and looking at its impact on the development of asthma. Now, both of these cohorts, the US and European, are two cohorts. I'm actually collaborate with this group, and I'm a part of this group, had a pretty similar study design. Now, this was really asking the question, if we have sufficient vitamin D during pregnancy, can that have a long-term effect on the prevention of asthma? Because a lot of our immune system is created in utero. So we took these individuals, found them very early, 10 to 18 weeks pregnant. We randomized them, good study design, to either the regular amount of vitamin D or an astronomically high level of vitamin D. Followed the individuals up, they delivered. We followed them through age 3 and looked at all these different measurements related to asthma and allergy. Now, this was done for both studies. This VDART clinical trial that I was involved with had about 781 individuals. There's a few differences here. They had 400 was the placebo group because ethically you cannot give someone 0 during pregnancy. That's what's recommended by the FDA now. The treatment group had 4,000 more IU, and we were recruited, like I said, 10 to 18 weeks. This COPSAC cohort was a little bit smaller, and they randomized. The randomization group was a little bit less. Women were recruited later in pregnancy, but were also assessed by age 3. So I want to point out here, in contrast to our autism study, we have high sample size. They're good randomized controlled clinical trials. They were double-blind. They had very good follow-up. They had great exclusion-exclusion criteria, and they were also ethnically diverse, so we can infer to multiple ethnic groups. So here is the paper that's hot off the press. I think it's actually in press now online. And this is actually the proof of it. And I want to look at a few things a little closer here. So here we want to zoom in and look at this. So this is one of the two studies. And here we have a measurement. Now think, these individuals are 3 years old. At 3 years old, how well can you assess whether someone has asthma? Sort of, but not really, really well. But we can assess if kids are wheezing. That you can hear, and the doctor can assess it. It's a much more reliable measure. And we basically look to see how those who are on the control arm, how much persistent wheezing they had compared to those who had vitamin D supplementation. And as we see, there is a difference, but statistically, it was not significant. When we look here at the measure of vitamin D at the very beginning of the clinical trial in the mother, so the question here would be like, really early on, how important is the vitamin D that the fetus is getting in relation to asthma? We also see a similar trend where those with really low vitamin D have more persistent wheeze versus those in the middle and upper quartiles. And this was significant. But sadly, when we look at these results and we look at them adjusted, although there is a visible difference, these differences were not statistically significant. Okay, so I'm going to pause here and ask the question, so when you see this, what do you think? And I'm curious to hear some comments. What do you think? Does it look to you like there might be something there? Like, no, there's not something there? What's going on?
Not sure. There's a hand for that.
Yeah?
Well, you're mentioning the idea that 30% approximately individuals might be deficient.
Yeah.
in vitamin D. So first thought that came into my head is perhaps you're just restoring function, so naturally performance might just be better.
Mm-hmm. Go ahead.
As someone with asthma, makes me wish these studies have been done long ago. If that's true, I wish my mom had more vitamin D.
Oh yeah. And trust me, as I was going through these clinical trials, my boss was saying to me, you better be taking your vitamin D. So, go ahead.
So you're talking about correlation, but you're stopping short of causation, is that right?
That's correct. And actually, this one slide is out of order. So I want to go to this slide. So what I will say as a statistician looking at these data, actually, I'm going to pick on James O'Malley. Where did he go? James, since we went to grad school together, can you tell me what you think about this?
Well, it could be that there is a difference. You just don't have enough sample size yet, or you've not fully long enough.
Ding, 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 ding. I knew one of my friends here would pull me through. This is exactly, when I look at these data, I say, okay, there's absolutely a trend here. When you look at these lines, they are consistent. They're actually remarkably consistent in that those with lower vitamin D have more prevalence of asthma, but it's not significant. So my first thought is this is probably underpowered because it's showing a result, but it's not quite there. And other things I think of are the kids are 3 and you're looking at persistent asthma. What about a much more valid measurement of asthma? Let's follow them to age 6. Age 6, you can really define whether someone's asthmatic or not much better because it's much clearer. So when I look at this, I say there's lack of statistical power, and we probably need to follow them up to get a more valid diagnosis of asthma. And the slide's out of order. And this is something I want to point out. So this is now a meta-analysis combining the data from both of those trials together. So now we're improving statistical power. And what do we see here? We see, if you look at this, the interpretation of this number is this is a hazard ratio. And this says that high vitamin D reduces your risk of asthma by 25%. And it's statistically significant with a p-value of 0.01. So here's an example where if you looked at maybe this one paper here, it would be negative. You combine it with a second paper that has a very similar study design, and you actually see the effect. So what conclusions can we have here? You know, in this case, the initial studies were both well-designed and valid, but they were underpowered, and a subsequent combination of both trials together improves our ability to find the result, and we can conclude that it looks like increased vitamin D supplementation during pregnancy is associated with a 25% decreased risk in wheezing by age 3, and most likely even stronger when we look at asthma. So let's add, why do scientific findings change? Here's some more reasons. Lack of statistical power. Using a very stringent p-value cutoff, although I think is good in a lot of cases, if we just disregard studies based on p-value, we won't find things. Another thing I want to point out here is this concept of difference in study design. In one study, we only had a supplementation of 2,400 IU, and the other one was 4,400 IU. If you have different levels of supplementation, one may show an effect while the other doesn't. So that's a very subtle difference, but it may have a dramatic effect on the conclusions. Here are a bunch of other studies that would generally fall under the same headline, but actually ask slightly different questions. You know, in the middle of an asthma attack, is taking vitamin D going to help you? Probably not. It's not a treatment for acute asthma. So adding another one to this list is different questions around the same topic are obviously going to have different answers. Okay, so going on to another headline here. Should you or should you not take hormone replacement therapy? And when I was sitting in my qualifying exam during my PhD, I knew this was going to be one of the questions that they asked something about because this had just come out in the news the fall before. And let me tell you a little bit about this. Well, hormone replacement therapy has been used to augment people who have either had a hysterectomy or during menopause when there's all these symptoms, as we can see illustrated here by the flashing mom with sweats. Great things about natural estrogen is we know it's protective against heart disease. And we know this because women are protected against heart disease pretty much until menopause, significantly when compared to men. So a logical biological hypothesis is, hey, if natural estrogen protects against heart disease, you know, wouldn't hormone replacement therapy be great because it's just going to continue to help protect you against heart disease? So that's one of the questions that was asked. Here's just some more of the physical symptoms of hormone replacement therapy, of menopause. So this study was really addressed in a well-designed way through the Women's Health Initiative. In 1991, they launched this clinical trial that took 161,000 postmenopausal women, and they randomized them to different treatments of hormone replacement therapy. And they looked at several outcomes after a period of time, and one of them was protection against heart disease. But the results were stunning. This really shocked the medical community because in 2002, they stopped the trial early. And why did they stop it early? Because it raised serious question about the benefits of hormone replacement therapy and actually suggested that there were significant, important increased risks that women should know about. And what were these risks? Well, there were many. When women were on estrogen plus progesterone, they had increased risk of heart disease, not decreased risk. Also increased risk of several other things, like pulmonary embolism. You see all these in red. Some of these were statistically significant, some were not. But enough for us to put a break on this trial and say, hey, we need to assess this. Furthermore, the medical community really was shocked by this. And here are some quotes that I thought really echo the passion and the surprise that people felt. Number one, this is the biggest bombshell that ever hit in my 30-something years in the menopause area. Number 2, the study results will have a tremendous effect. They overturn something we've been preaching as preventative for all these years. All the women need to be reevaluated and recounseled. Last one is the one I probably like the best. How did it come to be that one of the most commonly prescribed hormone replacement therapy drugs in the country that was used for decades in so many millions of women before the long-term effects of this were ever studied systematically? So here is an example where, really without any scientific research, we just sort of inferred the hypothesis that, hey, if natural estrogen is going to protect against heart disease, let's do hormone replacement therapy because it's also going to protect. And what happened was our scientific finding changed because of answer 6, surprising biology. And, as scientists, this type of thing happens over and over again. There are things, we have a hypothesis, it's driven by our biological understanding, and sometimes it surprises us. Okay, moving on to our third and final topic here. I am passionate about many things. I'm passionate about my family. I am also passionate about running. So, okay, I did steal my proof here. But here's me running the Vermont Marathon last May, and on one of my runs, one of my friends said to me, hey, I had someone send me an article on why running marathons is bad for your health. And I sat there and I was like, what? Are you kidding? So here is the article, which I then looked up. Is running marathons bad for your health? And then even below, here is a newspaper quote, which I love. Fast running is as deadly as sitting on the couch, scientists find. What? Okay, so I should sit on the couch and eat potato chips, and that's going to be better or the same for me as running outside. Okay, so I was just perplexed by this and had to look into this one further. So here's some article excerpts. And these are in the same article, but they're two contrasting comments. Okay, number 1, and here are there's validity in one of this, and I'm going to now summarize several of the articles that I found. And this top point, I think, is the most valid, and the thing that needs to be considered the most. The research showed that high-intensity exercise sessions lasting longer than 1 to 2 hours can overload the heart, and after years of excessive exercise, thickening of heart tissue may develop. In some cases, and I'm sure these are very rare, the scar tissue can even create the possibility of dangerous irregular heartbeats and sudden death. Now, they have a point here, and I can see that this could potentially happen. And I can actually see physiology that after running something like a marathon, you have systematic inflammation in your body that may not be good for the next 1 or 2 days. However, I also want to say in the same article, we see what's written below, which directly contradicts this. When you look at the Journal of American College of Cardiology, they followed 55,000 people aged 18 to 100 after 15 years and found out that runners lived an average of 3 years longer than people who didn't run at all. And, failing to get any daily physical activity is not good, and running or even just walking a modest amount a few times a week is still associated with solid health benefits. So here in the same article, they have 2 points, yet there's headlines like this: is running marathons bad for your health? And running is as deadly as sitting on the couch, scientists find. So I want to go and look at an epidemiologist who commented about this in Runner's World magazine, and I was really happy to see this comment because this really drove home the point. So in this article that talked about the supposed dangers of running too much, they looked at all these different measures of running. And they said they noticed things like, hey, when you look at the proportion of people who, and so here we have number of individuals in these different categories. So like people who run over 4 hours a week, there were 51 of them. 50 of them. And of that 50, they followed these people 10 years, and they found that 1 died. So that's how you look at all these. So frequency of jogging, there were 413 people in this category, and 128 of them died. Actually, sorry, I think the total is the total here. So 51, where of those 51, 50 lived and 1 died. So here it's 413 plus 128 is the total, and the 128 died after these follow-ups. But they're making these claims on different quantities of running, different jogging paces, and they're saying things like, hey, those who run fast, are just as likely to die as those who run slow. But then they're also looking at things like, equating those, this group where there's one person who died with other groups up here that have much larger numbers. And down here is probably one of the worst cases where we have 5, 7, and 5. And my point is, when any type of statistical analyses were done on this, none of the p-values were significant because there just wasn't the data there to actually conclude anything. Yet they're taking these very small numbers and making these very, very controversial conclusions like fast running is as deadly as sitting on the couch. So when we're looking at these things we see in the news, my conclusion here is look it up. If you're thinking it sounds a little crazy, look it up, because if it does sound a little crazy, the inferences are probably not done based on solid statistics. And I would encourage things like this by looking at solid resources where we can look at data and they come up with conclusions like running for health, even a little bit is good, but a little more is probably better. Now, another thing I want to point out is when we're looking at all these things, it's not just looking, often the media has one big generalization when it might be better to make very specific hypotheses, because that's what good studies do. So rather than saying, running is going to kill you, or running is bad, let's be focused on our hypothesis and say, is running bad on your knees, and how are you going to measure that running is actually bad on your knees? Or is running bad on your heart? So are we measuring the thickening that may occur in the heart? Are we measuring heart disease? Is running bad, meaning is running in bad weather dangerous for your health? Does running cause inflammation? So my point here is good science always has very specific, well thought out, defined hypotheses. And often the media just sort of generalizes very specific, honed tests and hypotheses into these generalizations. So my last one here is, why do scientific findings change? Answer number 7. Generic conclusions to specific hypotheses and questions that have been asked, that generic conclusion is not merited. So in summary here, I want to just look and reflect on what we've talked about. When you're looking at these things in the news and Katie Couric says something and you're just sitting back there thinking, I'm not quite sure, Katie. I don't know. It doesn't sound right. Use the statistical knowledge you've learned and rely on the original manuscripts. Go back into the details if you're really that curious and see what the science says, because then you can make a valid judgment on your own. And one thing I always like to say is, if true, scientific research will be validated over and over again. And I really rely on that in terms of my hypotheses and my inferences that I make. When we find something and it looks really interesting, I always hope that I'm going to be able to validate it in another study or another way to really conclude that that scientific hypothesis is true. So recognize that conclusions also often depend on the specific study design and the specific question being asked. So if you're looking at a randomized trial and one randomizes people to just a small level of intervention and one is a dramatic intervention, the conclusions of those two things may not be the same. And conclusions are often made definitive based on a p-value of 0.05, but sometimes those studies may be underpowered, and really to get to the truth, you may need to combine studies together or increase the sample size or do something to really see what the real findings are. And really, the concept here is be a critical thinker. So with that, I just want to thank you for having me here, acknowledge all of these people who are a great part of my scientific community and have helped me in my research. And I'll open it up to comments and questions.
5%, right? Is there something?
Yeah.
Does that mean that out of 20 studies, I can expect one to be wrong?
That's correct. Yep. And actually, in the scientific community, 0.05 is really the default. But I always sort of chuckle because, exactly what you said, 5%. Actually, let me step back. A p-value is, when you look at the normal distribution, the probability of being in the 5 percentile or more extreme. So actually, it's not exactly what you're saying, but it's just a probability that you are in, what you are observing is in the top 5% or more extreme. So that's how I would define it. Under a null hypothesis. So it's not exactly what you're saying, but I think that one thing we need to think of is often a p-value is not really the best measure. When you look at data, I like to look at effect size. One of the things I love to point out is when you think of something like the study of autism, when you have a sample size of 10 million, that could end up, any study that has a huge sample size, if there's even a tiny, tiny difference from the null hypothesis, because you've now shrunk the variance down really small, you could detect a very tiny difference that clinically may not be meaningful. So sometimes it's best to also look at the effect size in addition to the p-value. James?
Yeah, great talk, Jessica. So I think I could probably add a few more of the questions. But one thing I thought you might have mentioned is the concept of publication bias. And another point is that so often randomized trials, especially human randomized trials, are done in very selective populations. People who have serious cases of the illness at hand, but are otherwise very healthy. And then when the drug or whatever gets approved, years later we find out that this bigger population that gets access to the drug, it doesn't work so well on average on them. And thirdly, the fact that because of financial pressures, typically trials do not have typically very long follow-up, and sometimes things happen down the line that can cause concerns or black box warnings and flip the evidence. Right,
all great points. Publication bias, often when we do studies, you can actually assess publication bias, which is a great point. I think, the concept of often you have to look at the context of the clinical trial and say, can we infer this to the general population? So, for example, the Nurses' Health Study, which is a great cohort, but it's a cohort of educated women who may be more healthy than the general population. Can we infer what we see in healthy nurses to a general population that's arguably less healthy? In things like that. Yes?
So just kind of looking at statistics in medicine, there was some recent stuff released about how processed meats can cause cancer and they're bad for you. So could you talk a little bit about that study, whether you think that it's legitimate and whether it also goes over to red meat and whether you personally have stopped eating meat because of that?
So I will be the first to say I'd have to look at the study in more detail, but I think, honestly, of some of my favorite studies to look at are nutritional studies, and ones that are done well really have some overwhelmingly convincing results. And I really like, there was a study done years ago that was fascinating to me because it showed the increased risk of different types of fat. So if you substitute olive oil with butter versus trans fat, and what it showed was olive oil, and looking at this with regard to heart disease, olive oil was protective the more you had against heart disease. Butter was bad, but nowhere near as bad as trans fat. So I read that article and I said to myself, note to self, when I make my chocolate chip cookies, I'm using butter and not trans fat, because I like butter and it only has a little risk, whereas trans fat has a lot. So I'd have to look at the study more, but I think, that's what I would say.
It seems like insufficient power is kind of a very expensive, mistake to make in a big study
That is a fantastic question. And the key thing is, because I write a lot of grants and I have to do those power calculations in every grant, when you write the power calculation section of a grant, you're always assuming a certain effect size that you see. And I think that's one of the key things that often what we expect, like, vitamin D is going to have a 50% reduction on asthma. Well, if it's only 25% instead of 50, that's going to dramatically change how many people you need to have in your study in order to see the result. This particularly comes up in genetics, because when genetics started years ago, they thought, we're going to look at just a few genes and we're going to see these huge relationships to disease, and with just like 500 people. Well, now, 15 years later, because I've been in the field, what we've realized is we don't need 500 people, we need about 100,000 to actually look at that. And I can tell you, 15 years ago, we were writing power calculations saying that, we have enough power with 500 people, and now you need like 50,000. So.
Yeah, I was wondering about the two asthma studies, how you combine them, because if I noticed correctly, you used two different ways of measuring whether someone had asthma or not.
Right.
So how did you combine those two studies?
So the good thing is, one was persistent wheeze and the other was asthma wheeze. And when you do these type of studies, actually they were reasonably cohesive. But you can do different things like random effects models, which is a typical way that a meta-analysis is done, and that's what was used in that study. But it's a good point that when you're combining studies together, meta-analyzing multiple data types is not necessarily, it's not as ideal as you might want.
For the running will kill you article here, do you happen to know or tell us what the original publication was? Which editor was lazy to put that out?
Well, so I know that so it's interesting because the original publication, this group published the same data in two journals, and one was the American Journal of Epidemiology, and the other was the American Journal of Cardiology, some type of cardiology. So I'm familiar with the American Journal of Epidemiology, and that's quite a solid journal. Different journals have different levels of prestige and rigor to get into. So their conclusions in the American Journal of Epidemiology were actually different than their conclusions in this other less profile journal. And it probably had to do with the peer review process. So.
One last question. So this is kind of a question about, so when you see studies out there about any given topic that would convince you or try to convince you to change your behavior, I guess a big problem in the U.S. public or maybe the global public is that people typically just kind of go with their opinion and not really informed statistical or scientific analysis. And a lot of people just don't have that background. I was wondering kind of how can the average American, besides studying statistics, which would be awesome if everyone did, but they won't, how can people be better equipped to make informed decisions about their lives? And like, how do you go about, if you see a study, like, do you actually change your behavior based on that? And what's enough to convince you to not eat something if it's actually bad for you?
So that's a great question. I will say when it comes to desserts, no study really convinces me either way I'm going to have desserts. But when it comes to certain other things, like because I've seen a lot of the data, it's hard to answer that. But like for the lay person, I think it's important to look and see things that are validated over and over again. I will say absolutely, I changed my eating habits when I read that article about trans fat, butter, and olive oil. And I always try to substitute olive oil for butter when it comes to savory eating, or even in desserts, I'll do like a canola oil for butter. Another one that I absolutely changed was when I saw the overwhelming evidence about wild salmon versus farm-caught salmon, because the impact and the study was just, when I saw the actual results, was just tremendously overwhelming. So wild salmon is better, absolutely. Another one is some of the different works I've seen on like omega-3 fatty acids. So the fats found in things like salmon and the health benefits of that, particularly in development, brain development, and so on and so forth. So I absolutely do, but I'm also in a community where these things are talked about all the time. And I'll just say one last point on this that I found fascinating. When I was doing my doctoral work, and I was in at the School of Public Health, I took a nutritional epidemiology class by probably the world's expert in nutritional epidemiology. And one of the things he said to me that was so striking is, this is years ago, and he was talking about milk and the fact that there are hormone levels in milk and they're just out of control, and the FDA really should regulate that better. Now, that was in 2000, and now it's 2015, and we're hearing about this over and over again. And there's several things he said about even the way what's recommended on a federal level, unfortunately, gets impacted by special interest groups like the beef industry and the dairy industry and so on and so forth. So I think that's something that I think we need to be careful of.
Jessica, thank you. I do have to say I feel the urge to go out and run and eat a little healthier. We actually did decide for next week we're going to start having fruit and yogurt here as well. Very timely, but thank you.
Thanks.